Hello, and thank you all for coming to today's lecture, which is on how low-level CO impacts the adult and developing brain. My name is Mari Herigstad. I lead the Sheffield CO Research Group, and we have been given the opportunity to talk to you about our ongoing research, which Court is funding. They've given me the task of hosting, so I'm hoping this goes well, but if it doesn't, it's entirely my fault. As many of you will have noticed, we have a lot of speakers today. And so after discussions with the core team, we have agreed to omit the traditional Q&As to avoid overload, but we'd very much like to answer your questions and hear your comments. So instead, please can you add your questions in the chat or by email to lauren@coresearchtrust.org, and we will answer these as a team in a blog post after the talk. That way, I hope we won't exhaust you entirely, and it might also mean that we have a fighting chance of not missing any questions. So I'm just going to see if I can start sharing my screen. Share. Apologies. So I'm hoping you're all able to see this. I wanted to start this by giving an overview of who we are up here in Sheffield. Here are most of us and the wider team. Not all will speak. I'm sure you'll be happy to hear. We have our fantastic PhDs and master students here, without whom we'd be absolutely stuck. That's Simon Clark, who might be a familiar face to many of you, Lucy Wilson, Josh Dorrance, Megan Hurd, Trifin Gumbo, and Tabitha Stutley. On this other side, we have myself and my collaborator, Patrick Stafford, and our great research fellows, so that's research fellow Duncan Garner, and our visiting researcher, Elsie Place. We're fortunate here at Sheffield Hallam to be really, truly multidisciplinary. So we have used this to our advantage and brought in several researchers to the CO course, each from their area of expertise. So we have Inesh Ramos-Harrington, who will pair her work on MS with CO. We've got Samrain Ahmed, who will be talking about her research into cancer and how it links with CO. Today, we've also got working with Nick Peake on extracellular signaling and CO, and with Dr. Stephen Megbo on sleep apnea and CO. So all of these people are involved in the wider CO work that we do up here. We're fortunate to be supported by Dr. Celine Swilhall, who's a senior technical specialist, without which we'd probably get nothing done. We also have, we're fortunate to have a really good range of good collaborator, chief among which is Maricha Placek, who is a developmental biologist and is helping us with the developmental stuff that we're doing. Chris Morris, who gave a fantastic talk earlier this year to this lecture series, and if you haven't seen it yet, I suggest you go and have a look at the recording. It was great. And also Liam Bridge of Manchester, we're working with on cardiac development. So today, you'll hear from the following members of the team. So Simon will introduce what we're doing here, and we'll follow by a section on development, so the developing embryo by Elsie and Duncan, followed by a video from our social media lead, Trifin Gumbo, who will talk about her work on white blood cells. After that, we'll have a CO and disease segment, which is with collaborators Samrain Ahmed and Inesh Ramos-Harrington. And following that, we'll talk about the adult sort of side of things, and Megan will talk about her work on adult brain cells, and I'll round it off by talking about what we do with human experiments. So given that we don't have much time, and without any further ado, I'll give the floor to Simon.
Hi everyone, hope you can see me. Some of you will know me, I'm Simon Clark. I have been a doctor in the past, I've now retired, and I'm bugging these guys here and collaborating with their research and doing a PhD here. So I'm here really to introduce the background so that no one else has to do it. Otherwise you're going to have, umpteen dozen people saying the same thing. So what is CO? It's colourless, it's odourless, you can't smell it, you can't see it. It's the product of incomplete combustion of fossil fuels. It's probably the leading cause of accidental death by poisoning and one of the leading causes of intentional causes, but we don't know about the accidental deaths because we don't routinely at the moment test for it in autopsies. And because there's no characteristic toxidrome, the clinicians, probably me included in that, have missed it quite a lot. It is probably a significant burden of morbidity as well. There's certainly been some research done in Taiwan looking at dementia and the links of dementia to CO. And of course, my particular interest is links to congenital heart defects in the newborn. Long history. I'm not going to take too much onto this, apart from the fact, as you can see here, we've known for over 2 millennia that coal fumes are bad for you. And since the 1770s, so about 250 years, that's when CO was first detected. And subsequent to that, we've identified as CO as the thing the main component in coal fumes that is really bad for you. Interestingly, we knew a lot about the mechanism of toxicity, but in the 1940s, 1950s, this chap, Sjögren, actually discovered the fact that CO is formed endogenously in the body. And it does have physiological effects, particularly on blood vessels and possibly on a mediation of inflammation. I'm really going to rush through because I'm assuming most of the people here have got, in the audience have got an interest in CO and know it works. It binds. It loves heme, so it binds to hemoglobin. It causes, in significant enough amounts, it causes tissue hypoxia. It also reduces the store of oxygen in muscle by binding to myoglobin, and it affects the mitochondrial cytochromes. This is my area that I will be researching, particularly complex 4 or cytochrome C oxidase, and that leads to apoptosis, oxidant stress, oxidative stress. Interesting though, the endogenous breakdown of CO, the endogenous formation of CO is from the breakdown of heme. One heme molecule broken down forms one molecule of CO. And as I've said, it does have physiological functions. So really importantly, and this is, I think, a key part of what the group are doing, is answering that question of what dose does CO become toxic? Does it go from a physiological agent to a toxic agent? Second question, are ambient levels that we breathe out here in a city or near roads, are they dangerous? And are there any particular vulnerable groups, the unborn child, the elderly, premorbid conditions? And obviously from the all-party parliamentary CO group, it's a real key, the effects on the fetus and other vulnerable groups, but the fetus particularly has become an issue for policymakers, and they're keen that research is done. Epidemiological data, there's plenty of it for fetal effects, but it's all to do with smoking and pollution. Is CO is not the only potential toxin in either of those routes. So we're here to try and determine what the role of CO is. The fetal effects, a lot of the work I've looked at has been on rodent studies, but the problem is with that, if you put a pregnant rat into a cage, it gets stressed and it stops eating as much, and there are effects on the placenta. Now, the effects that have been seen on the fetus could be a result of these, and that's one of the things we're worrying about, and that's why we are going to be talking quite a lot about, as well as cells, but our in vivo model is going to be in the chick embryo, because the chick obviously has its own little capsule. It sits in called an egg, and the effects of CO are going to be on the embryo only. There's no placenta. So those are the two models we're going to be looking at. We do have, from a study that was done 2021, that there is potential that actually there's some evidence that fetal hearts, even at very, very low levels, that's down to 1 part per million, really low levels, is associated with an increase in heart size in the chick model. That's it, I'm done. I am now going to hand over to Elsie and to then my colleagues after that, and thank you for listening.
Thank you, Simon. So hi, I'm Elsie. I joined Mari's group last year, and it's my role to kind of help get this chicken brain project off the ground. So for the first few slides, I'm going to give you even more background, but a little bit more specifically on carbon monoxide and brains. And later on, I'll share you I'll share a couple of selected results with you. So as far as carbon monoxide and brain development, yeah, there have been a number of studies since about the 1980s that have made a start on this problem and have reported CO-induced changes in the offspring of pregnant mice treated with CO, typically in the range of sort of 75 to 300 ppm. And the kind of changes they've detected are changes in brain chemicals, so neurotransmitters like dopamine and serotonin, and that range of CO doses was kind of optimized to achieve carboxyhemoglobin values similar to those seen in heavy smokers, or well, similar and above in effect. So the dose is obviously a bit higher than we're interested in. Being sort of fairly old studies at this point, they weren't able to take advantage of modern techniques, so they're fairly crude in their methods. There's been a nice paper recently, this Trentini et al. Paper in 2015, however, which showed that at a dose of 150 ppm, the migration of brain cells into the cortex was impaired, and they showed some perturbation in biological signaling pathways. So there is some background to this, but not a lot of detail done. We're using the chicken, of course. These studies were all in rodents. And in the chicken embryo, there have been kind of general studies on toxicity. So looking at growth and viability. This study by Alexander identified 1,000 ppm as being toxic. And this other earlier study showed that viability was reduced at 425 ppm, but not at 100, and there was nothing in between. And interestingly, they also identified in the same work that there seemed to be a critical period of sensitivity to CO effects between about day 8 of embryonic development and day 14. And as Simon's just mentioned, in Mari's group more recently, in the heart, it's been shown that much lower levels of CO can increase the ventricular wall thickness, so single digits to low double-digit concentrations. So to begin to fill in some of these gaps in the literature, my initial aim was to look specifically at brain growth, not whole embryo growth, but brain growth, and to establish what CO levels we detect an effect on brain volume and brain dimensions, and to use this as a crude measure of toxicity. So it is only a crude measure, and it does mean for this first part of the project that I'm using higher doses than we intend to explore down the line and in Duncan's work, which I'll hand over to shortly. So next slide, please, Mari. So for those of you who haven't seen embryonic chicken brains before, I've included these pictures that demonstrate some of the ways that we can image and analyze these developing brains. So in the top row, there's a series of images from early to late, where the embryos have been stained using antibodies or a technique called HCR to look at where the different genes are active. So in the top left, that's what we call a stage 10 embryo. And at this early stage, the neural tube is just kind of like a cylinder with a little swelling at the front. So what you're looking at in these images are embryos that have been cut through the middle of the head, and you're looking at the right-hand side from the inside. So that left one is the stage 10, and so the left is the front, and in the next two pictures you can see that this area of the brain just grows and becomes much more complex. And the last picture is a 3D render of a couple of genes expressed inside a day 10 brain that we can image in 3D using a special microscope. And then on the bottom, we've got a micro CT scan of a chicken brain at day 10. So they're fairly well developed at this point. You can easily identify the main regions, and just for your comparison, there's a human brain in the middle, and there were some quite sort of obvious differences in the proportions of things. So that little blue area in the human brain is the midbrain, which in the chicken is that large lobe that you can see in the middle of the picture. So sort of in the middle of the brain. But despite these really sort of obvious differences, there's a high level of conservation. So they're really useful nonetheless to learn about our biology. So in a minute, I'm going to show you a short video of early forebrain development, going from stage 10 to stage 20, as per this bottom right image. So in case you're interested, and if you know anything about brains, the regions of the animated brain are coloured according to these labels, and this video will hopefully kind of illustrate how the proportions of the brain change as the retina of the eye grows out from the brain. So if we can go to the next slide, please. So what you're first going to see here is a stage 10 embryo with lots of coloured dots on it, and they're round dots. And as the brain develops, these dots will change their shapes as they grow, and that is going to show you what direction the tissue's growing in. So you can see from the variety of these shapes that develop from those dots that growth is not uniform across the brain, and actually there's all this growth that goes in the direction of the optic stalk, in that where the arrows are pointing to, which causes the retina to be extruded from the brain. So if we maybe skip a little bit to about a minute. Yeah, so this shows the same kind of thing. If you keep your eye on that large opening to the retina with the circle around it, you can see that it grows narrower and narrower as the retina grows out and the optic stalk develops. And meanwhile, all the other regions around it become proportionately much, much bigger, especially the telencephalon there in purple. So if we go to about a minute 20, we might almost, 150. No, this is good, sorry. Okay, so where are we on? Okay, yeah, so this is going to show us the same thing from the outside. So similar to before, so this is now an outside view. These dots are going to stretch as the embryo grows. So the retina is just at the outside of that swelling there, and it's going to show you how the tissue stretches along the optic stalk, while some of the brain regions grow much larger than others, and the brain and the retina grows out into the eye. So hopefully that's kind of cool and interesting. And now I'm going to move on to slide, please, Mari. I'm going to, so start giving you an idea of how I've been approaching this question of carbon monoxide toxicity on brain growth. And essentially, I'm looking at three main measures at the moment. So I'm looking at oxygen consumption of the embryos, at the brain growth, so the volumes of the brains, and cell death at a range of different CO concentrations. So this box on the right is these are special incubation boxes, which are airtight, and they've got these valves to which you can attach tubing, and that means you can pump the air out and you can replace it with air that's got carbon monoxide in it. So we do this once or twice a day to replenish the air in the box, and the chicken eggs go in there. Click, please. Okay, so based on this very preliminary observation on day 3 chicken embryos, I decided to investigate a range up to 400 ppm CO, and that was because, as you can see from these embryo heads, when I went up to 800, the embryos were very small. That was a highly toxic dose, but at 400, I thought I was seeing marginal effects on growth and a slight developmental delay. So that looked like an interesting dose to carry forward. Next, please. Therefore, I chose doses of 0, 40, 120, 200 and 400 ppm. And this is just some example data from handheld loggers that we can leave inside that show we get quite accurate targeting of the doses we want. So the next slide. So for this experiment, I'm going to show you some results from a 5-day experiment where I imaged the embryos by micro-CT. This sequence of images gives you an idea of some of the things I measured in the experiment. So on the left, that is the reconstructed micro-CT of the embryo that I scanned, and then the internal structure is highlighted in the next image along with this cyan object, which represents the brain ventricles from the midbrain and the forebrain. So the ventricles, this is the space inside the brain, and at these early embryonic stages, it's very large in relation to the total brain size. This was something I used as a proxy for brain volume, and then using a slightly more elaborate process on the right of drawing round the neural tube and selecting the tissue inside, I was able to get a measure of tissue volume. So the results are just on the next slide. Thank you. So what I found when I measured these parameters was that only at a dose of 400 ppm did I see a change in brain size. So I measured a number of distances within the brain, like eye spacing and floor plate to optic midline, and the result was consistent for all the results. So in the two measures I mentioned, so the graph on the left, I've got all of the doses here. So this is for ventricular volume, and as you can see, that is significantly lower in the 400 ppm dose group, but it seems to be kind of a threshold for that effect. And the brain tissue volume measurements are not quite complete, so I've just shown you the 200 dose that shows you that in this measure too, we don't reach significance at 200 ppm, but all of those other metrics were significant at 400. So this is a very helpful result for a few reasons. So this 400 ppm dose corresponds to the toxic dose reported in the earlier literature, where viability was affected in that case. And then furthermore, this relatively recent study showed that mouse fetal growth was reduced at 400 ppm, but not at 300. So despite this mouse study being performed in a placental species, we seem to have got maybe a comparable result, a comparable threshold. So that starts to suggest that our experimental system with the chicken eggs in the boxes may give similar effects at comparable doses to mammalian systems, which is not a given until we've confirmed that. So we finally, now we've kind of got a reference point, a high reference point that we know leads to kind of overt toxicity in our system. So we have this top dose of 400 ppm, where we know this restricts brain growth and starts to compromise viability. So that leads to the next slide. So now that I've got a dose of CO that I know is overtly toxic, my strategy currently is to use this high dose to look for changes in brains that might be harder to spot if you only start by looking at a lower dose. So once I identify a change at a high dose, I'll then be able to look at whether something similar is happening at lower doses. It makes it a little bit easier to start off with. And for the next experiments, I'm working on embryos taken to day 10 of development. And I just wanted to show you this little snippet of data because it shows something a bit mysterious and interesting, which is that I measured the overnight oxygen consumption of my eggs in the boxes, which I was incubating 2 to a box in 0 and 400 ppm CO only. So up until day 8, as you can see from these lines, the oxygen consumption was identical between the 0 ppm groups, which are the blue lines, and the orange 400 ppm group. So we're looking at day 8 there, between the blue and the orange. But from day 9, And definitely at day 10, a difference opened up between the two groups. And there was this divergence in oxygen consumption where it was higher in the air group. And there was also a spike in embryo death, which seemed to be occurring from about EA onwards. And we don't know why. We don't know why this is, but it's likely to be vascular or metabolic effects. And even where both eggs survived in a box, we still saw that the CO treated embryos are consuming less oxygen. So this seems to confirm what I said in the beginning about that earlier study identifying this period of increased sensitivity between day 8 and 14, but we don't know why that is. And it also raises the question of whether we might see effects on growth at lower doses at E10, which I'm going to explore by micro-CT. So finally, so I haven't talked about everything, but I'm currently looking into cell death in the day 10 chicken brains. And again, at the moment, I'm looking at 0 versus 400 ppm only, with a view to then seeing whether any changes that I might detect are also present at lower doses. And I'm planning another micro-CT experiment on day 10 brains, and this is quite a nice time point, so mainly because it's in this more sensitive period, but also we can do quite a nice detailed regional analysis at this point and compare and see whether different areas of the brain are differentially affected, whether some are more vulnerable than others or not. So to make a few concluding points, the incubation system that we have produces effects on growth that are comparable to those already reported in chicken and mouse. 400 ppm CO seems to be kind of a bit of a threshold for effects on growth and viability, growth of the brain and viability. And we think from about day 9 onwards, the chicken embryo is more sensitive to the effects of CO. So I'm going to hand over to Duncan now, who's going to talk a little bit more about oxygen and hypoxia.
Hi everyone, I'm Duncan. I'm a postdoc in Mari's lab, and I've been working on this project for about 6 months now. And I'm going to be sharing with you something that Elsie touched on, which is oxygen consumption and hypoxia in our chick embryo model. So just for those of you that aren't aware, hypoxia is a state of reduced or limited oxygen availability, which is of course detrimental to respiring organisms such as the chick embryo. Why is it relevant to our research of carbon monoxide? Well, as many of you know, it's the primary mechanism of carbon monoxide toxicity, especially at higher doses of carbon monoxide. But it's important to note that it's not the only mechanism of carbon monoxide toxicity, especially when we're looking at lower doses of CO. And so it's important for us as researchers to be able to delineate the hypoxia-dependent and hypoxia-independent mechanisms of toxicity. When we get down to the molecular and cellular level, there's actually conflicting evidence surrounding the mechanistic role of hypoxia signaling and hypoxia pathways in carbon monoxide toxicity. So it's really interesting for us to research this further and hopefully shed light on this mechanistic role. It's important to note as well that hypoxia does have a physiological role, especially in neurodevelopment specifically. So for example, in the chick embryo, the embryonic and fetal brain actually exists in a state of hypoxia that changes and fluctuates throughout the course of development. And it's actually believed that these changes drive signaling events and developmental events that are key and critical in physiological development. And finally, just to note, so Elsie showed our incubation and exposure methods with the incuboxes. Of course, incubating chick embryos in these airtight boxes, if you keep them sealed for too long, then there will be a hypoxic environment eventually. And so Elsie touched on a sort of mild hypoxic environment within those boxes after a 16-hour overnight incubation, and she showed her nice data. And my data on the right here shows a similar thing, just plotted as oxygen consumption per gram of live egg weight. So we can kind of use this as a readout of respiration rates in these chick embryos. And you can see, hopefully, that our embryos exposed to 0 ppm carbon monoxide, so our controls, seem to consume more oxygen compared to all of those exposed to carbon monoxide. And interestingly, there's no dose-dependent effect with the carbon monoxide there. But the real thing of note here is that, of course, as the embryos develop more and more, towards the later days of incubation, the oxygen consumption kind of skyrockets. And in line with what Elsie showed, we do start to see a mild hypoxia in the morning when we open these boxes up. And so that's something I wanted to look at further. And I wanted to look at the molecular and cellular level. So I started with this protein HIF1 alpha, or hypoxia-inducible factor 1 alpha. This is a protein that regulates the cellular response to hypoxia. And it does so in a sort of, in a rough manner. This schematic shows that hypoxia activates HIF1 alpha, allowing it to bind to DNA and induce gene expression, leading to cellular and tissue level changes such as increased blood flow, a decreased aerobic respiration, and increase in anaerobic respiration, all in an attempt to counteract the detrimental effects of that hypoxia. And it's worth noting at this point that this entire system, the proteins and the genes, is very highly conserved between species, it's including between humans and chicks. And so the chick embryo does remain a good model even at the cellular and molecular level here. Now, I mentioned that hypoxia activates HIF1 alpha. It actually does so by stabilizing the protein. And so under hypoxic conditions, we'd expect to see increased protein expression of HIF1 alpha. And protein expression is what I went on to look at. So my data is shown here on the right, and this top right graph is protein expression of HIF1 alpha in chick embryo brains collected in the morning, so under those hypoxic conditions within the incuboxes. And hopefully you can see that HIF1 alpha protein expression actually decreases as carbon monoxide concentration increases. And that effect is significant at 120 parts per million. And so that's kind of in line with what me and Elsie is showing, where there's decreased oxygen consumption, perhaps leading to a less hypoxic environment within the box, leading to less HIF1 alpha activation. So I then wanted to go on and examine whether carbon monoxide alone, without hypoxia, has any effects on HIF1 alpha expression. And so the data on the graph in the bottom shows just that. So this is a similar experiment, but the chick embryo brains are collected around midday after the boxes have been opened up in the morning to allow that hypoxia to dissipate, but the carbon monoxide is re-added. And at this point, although this is preliminary data, it looks like there's little to no difference between conditions in HIF1 alpha protein expression. And so it seems that carbon monoxide does not seem to be having an effect on HIF1 alpha protein levels, at least at the concentrations we're looking at. Now, it's worth noting that HIF1 alpha protein expression is not the be-all and end-all of hypoxia pathways, and it's important to also look at HIF1 alpha activity. And what does that look like? Well, HIF1 alpha is a transcription factor. As I said, it induces gene expression in its target gene pathways, and some of those are shown here. And so by examining the expression of these genes at the mRNA level, we can use that as a readout of HIF1 alpha activity. And so I've decided to look at two genes for now, LDHA and VEGF-A, which are included in anaerobic metabolism and angiogenesis pathways, respectively. And before I move on to talk about my data, I'd just like to mention that these genes outside of the hypoxia response pathways are actually also neuroprotective. So they have been shown to enhance survival of cells within the central nervous system, highlighting their importance in our research, both in the developmental model, but also in adult models. And so going to my data on the right, again, very preliminary, and these are chick embryo brains collected again around midday, so without that hypoxia, carbon monoxide only. It seems that there are no general trends and little to no differences in gene expression between conditions. And so it really looks like carbon monoxide is neither activating HIF1 alpha at the protein level or causing its activity to increase. So just in conclusion then, based on my very preliminary data, our incubation methods include a temporary mild hypoxic exposure that's somewhat unavoidable due to being overnight. And this is associated with a transient response in the chick embryo brain that dissipates when that hypoxic environment dissipates as well. Carbon monoxide exposure alongside that hypoxia may lead to a reduced HIF1 alpha response in the chick embryo brain. So there's a somewhat interesting interaction there. And finally, it appears that low-level carbon monoxide alone may not elicit a HIF1 alpha response either at the protein level or in activity levels in the chick embryo brain. So altogether, the sort of takeaway points from this are that although there is this brief hypoxic exposure during our incubation protocol, it does not seem to have any lasting effects, at least based on what we can see currently on the chick embryos. And also, any effects that we go on to identify with our low-level carbon monoxide exposures are likely to be independent of hypoxia because carbon monoxide at these levels is not causing any hypoxia response. That's everything from me. Thank you very much for listening. I'm going to hand over to Tryphene, who's got a video for us talking about the metabolic effects of carbon monoxide exposure in a blood cell model.
I think, do you want to give a wave just before we, hopefully this will work. Let's see.
Hi, my name is Tryphene. I hope you enjoy this video. The focus of my research is the effects of low levels of carbon monoxide on Jurkat cells. I have been looking at different levels from 0 to 10 parts per million and different durations of CO exposure. Through this, I've investigated metabolic activity and cell viability, and I hope to expand to investigate plasma samples in the near future. So why look at lower levels? Unless it's due to a specific lifestyle, work, or unsafe environment, some individuals are exposed to lower levels of carbon monoxide which aren't readily detectable via carbon monoxide alarms or diagnostic tools, and often people come into contact with these lower levels on a daily basis. As you know, carbon monoxide binds to hemoglobin and has an affinity 200 times greater with hemoglobin than oxygen. As a result, it causes cellular hypoxia as it lowers hemoglobin's ability to carry oxygen to tissues. So why Jurkat cells? Let's have a look. Jurkat cells are a motilized human T lymphocyte cell line, and these type of cells are a crucial part of white blood cells. They are frequently used in science research, specifically immunology and pathology. Jurkat cells are also non-invasive and readily available in our lab. This is a big advantage in experimental research when optimizing potential methods. Moreover, using this cell line, we can develop our current understanding of how environmental carbon monoxide exposure might suppress immune function. The cell viability and Alamo Blue assays both work hand in hand as they provide key insights into how CO affects Jurkat cells. This can inform us on if low-level CO hinders mitochondrial activity. My results thus far have demonstrated that the longer cells are exposed to CO, the more severe the impact can be. However, it has also shown that at these low levels, recovery of metabolic activity is possible, but only for short-term recovery. This suggests that low levels of exposure over a long duration otherwise known as chronic exposure, can still have a negative impact on health, or at least our immune cells. So given this interesting result, I'm very excited to expand this research into utilizing new modes of analysis such as confocal imaging, flow cytometry, and immunoassays, as well as exploring the effects of low-level CO within plasma samples collected during CO research. Thank you for watching the video, hope you enjoyed it.
I'd now like to just introduce Samray, who's going to talk a little bit about her work on CO and cancer.
Okay, good afternoon, everyone. So from all these very informative presentation after tracking nice video, so I'm going to delve into more solid science. So I hope I'm not going to be harsh on you. Okay, going in these coming slides, I'm going to explore with you what is the effect of carbon monoxide, low dose carbon monoxide, on neuronal derived cancer as well as breast cancer. So why these two types of cancer? What are the future directions? All this will be in the coming slides. Just before I start, I would like to disclose that the data in this presentation was produced by undergraduate students during their final year project, as well as technical staff. And having said that, that undergraduate is now involved in carbon monoxide research is actually shows how carbon monoxide is engaging our undergraduate students as well. So from previous presentation as well as from previous studies, it's very apparent that the main two systems that are affected by carbon monoxide, these are cardiovascular and nervous system in adult cells. But not only this, also this affects the embryos as well as the fetal growth. So because I'm cancer biologist, so definitely I'm interested to know whether carbon monoxide has any kind of mutagenic or carcinogenic effect. Having that said, according to the Department of Health and Human Services, as well as International Agency for Research on Cancer, as well as Environmental Protection Agency, they all identified carbon monoxide as non-cancer agent, so it's not causing cancer. Having that said, it indirectly might be involved in cancer. How? Because as Duncan said earlier, it has a role in sometimes inducing hypoxia, induced factor, sorry for the error here. And this one actually, when it's induced, it supports tumor growth and angiogenesis, something that cancer cells actually need. Not only this, according to Trifin, as videos as well, and Harris studies that carbon monoxide showed that there is a negative effect on the immune function. And when the immune system is negatively affected, also this gives the cancer opportunity to thrive. So all this give there might be a link between carbon monoxide and cancer. Why am I having neurological related cancer on board as well as breast cancer? Because they do align with my area of research. I have done some studies on neurological related cancer, as well as developing interest coming into the area of breast cancer. Interestingly, when I talk about neurological cancer, talking about glioblastoma, which is a malignant brain cancer, it is widely spread among different age groups, as well as the gender, female and males. However, it's more prevalent in adult males. And the other extracranial neurological cancer is neuroblastoma, that is mainly happening in pediatric patients. So there are some evidence, epidemiological evidence, that carbon monoxide exposure is linked with low survival rates in glioblastoma, which means there is a link between carbon monoxide and neurological related cancer. So when it comes to breast cancer, because I have my PhD as well as my master's student, we are looking at risk factors for breast cancer. And when me and Mari, we started to chat about carbon monoxide, I know that there is an epidemiological evidence saying that there is a more low risk for those who are exposed to carbon monoxide, they have lower risk to develop breast cancer. But when we talk about cancer, is it development or progress? So I think we need to make sure that we have a clear demarcation between these two. And when we talk about breast cancer, we mainly talk about the cells that are mainly gives us breast cancer, which are the ductal breast cells. And also we thought, me and Mari, within our chat, that could, if there is any effect on those cells, is that going to have somehow linked with lactation, because these are the ductal cells and they have another physiological role, which is lactation. And this is area that has barely been touched in research. So I'm going to share with you some preliminary data, and we consider the model we are using as well. So when we talk about carbon monoxide and its cellular effects, it's always the two things that we need to always think about is the dose of the carbon monoxide. And if it is low dose, in like very close to the endogenous levels, it has a protective and good role, physiological role, as Simon said earlier. However, when those doses are not tolerated by the system, this is when carbon monoxide is linked with toxicity and disease. And also what matters is duration and exposure, something that has been mentioned also by Duncan and Elsie. All this should also include what model we're actually considering to study the effect of carbon monoxide. So in this presentation, we are trying to compare the effect of low dose carbon monoxide on two different models, because when we deal with cells, we can either grow them in vitro in a 2D culture, when we grow the cells in a monolayer, or we grow them in 3D culture. So definitely they are quite different. So 3D is more like resembling and presenting how the tumors grow inside the body, and whatever is the 2D cells or 2D culture cells they are exposed to, it's always uniform exposure as well as a direct exposure. However, when we grow the cells in a 3D culture, we're not expecting the cells all to be exposed similarly to whatever treatment we are applying it for. So in this experimental, the main aim in here, we are testing how low dose carbon monoxide affect glioblastoma, neuroblastoma, and breast cancer cells, survival and proliferation. Why survival and proliferation? Whenever we think about cancer, the first hallmarks are sustainable survival as well as proliferation. And we wanted to check whether there are any differences between the 2D and 3D cultures and how they react to the exposure of carbon monoxide. So after the cells were established, the culture was established, the cells were incubated in incube boxes, the carbon monoxide and the control air conditions were settled, and then the cells were incubated in the incubator that has a regulated temperature as well as humidity. So these are preliminary data shown from undergraduate final project. So neuroblastoma cells, you can see collectively, so this is an outcome of a biological independent repeats. And you can see that carbon monoxide led to more non-dividing cells. So as if it kind of sequestered the cells in the cell cycle where they are not dividing anymore. And the same also has been observed with the glioblastoma cells. So it was the both. Although here these are representative figures, it might not necessarily reflecting this, but this is the average of many experiments that have been, more than 3 experiments that were performed for this average. So in 2D culture, so these are 2D cells, and this is how carbon monoxide affected them. So more non-dividing cells were observed with carbon monoxide treatment. When it came to 3D culture, we were surprised, and also it was an interesting observation because in 3D, the spheroids, so these are representing the 3D culture, instead of them subside in their growth, carbon monoxide actually slightly increased the growth of these spheroids. Same happened with in neuroblastoma as well as in glioblastoma cells. So which was interesting. So our conclusion from this set of preliminary data that the 10 part per million has anti-proliferative effect in 2D culture, because as we said, the cells were all uniformly as well as directly exposed to that dose of carbon monoxide. However, when it comes to the spheroids and the 3D culture, the same has not been observed. Rather, there was a slight increase in the growth. So our interpretation to this, so as we all know that these spheroids, the cells at the edge, they have more oxygen, for example, more oxygen and more nutrient exposure. However, when we come to the core of these spheroids, there is less oxygen, great concentration as well as nutrients. So this is why we have more hypoxic core. So the same applies to carbon monoxide. So the cells at the edge, they are having more of the carbon monoxide. When we go to the core, so there are carbon monoxide, but in low levels. And there is literature that's supporting that low dose of carbon monoxide actually leads to the increase of hypoxia-induced factor alpha that supports tumor growth. So this still needs a more confirmation, and this is what we are going to pursue in the future. So now, parallel to that, because I have a developing interest in breast cancer. So as I said before, with breast cancer, the story is a little bit different because carbon monoxide exposure has been linked to low risk of developing cancer. And scientists have made use of that by using carbon monoxide releasing molecules as well as low dose carbon monoxide in actually treating breast cancer and having an anti-cancer effect. But we wanted to see if this also applies in our models. So the same here, these are 2D cultured cells, and with the carbon monoxide. So in this time, we have challenged the cells more by having a wider range of carbon monoxide exposure. And you can see there was more death of cells that were grown in 2D culture in both the cell lines that we use from the breast cancer, because breast cancer has different molecular subtypes. So we wanted to see if the different molecular subtypes, they do have the same response to different range of carbon monoxide exposure. So there is a more death when cells exposed to carbon monoxide. When it came to 3D, these are the spheroids. So there also, to our surprise, there was less also death when compared to the treatment, to the control, which was also a surprise to us. So because we know, and as Duncan said before, carbon monoxide is linked to reactive oxygen species and to oxidative stress. And I think this also mentioned by Simon earlier. So we thought that carbon monoxide mediate this death in 2D culture via increasing reactive oxygen species or oxidative stress. But to our surprise, there was a decrease in reactive oxygen species. So this is something we would like to look forward to and try to investigate more. There are some few research talking about carbon monoxide as an antioxidant rather than a pro-oxidant. So in a summary, so carbon monoxide has more killing effect in 2D culture rather than 3D, and this is not related to reactive oxygen species generation. And this is something is kind of consistent to what we actually got with the glioblastoma and neuroblastoma cancer cell lines earlier, which means the model also matters. So it's not only the duration and the dose, the model also matters, and we are now looking forward to use the 3D model more and make more refinement in that and see how they represent the effect of carbon monoxide in cancer. So again, this is just a repetition that the 3D model, we are going to make more advancement and refinement to see how this represent the tumors, because when we talk about development of a tumor is different from having the tumor already on and whether carbon monoxide actually is going to support tumor growth using some of these models. So how we are going to take this in the future. So we are definitely going to look at some molecular and cellular mechanism through which carbon monoxide supports or maybe killing cancer cells in now currently we are trying with the help of Celine, we are trying to have a more established model using CAM assays, using the embryo, the chick embryos, and how we can have these spheroids in more in vivo mimicking environment to study how carbon monoxide affects tumor. And then we advance this knowledge by digging in with studying more the molecular and cellular events that happens with the carbon monoxide exposure. Since we have touched on breast cancer, and I did mention earlier about the ductal cells, and if they cause some death effect, there is the killing effect, we are actually thinking whether carbon monoxide affect the normal or non-cancerous breast cancer, breast ductal cells, and whether this has any effect in lactation or milk production, as well as the quality of the milk in lactating or breastfeeding women. With that, I'm going to finish my part. And so carbon monoxide is not only its effect in disease, is not only limited to cancer, it also expands to other diseases such as multiple sclerosis. And the coming video is from Dr. Inish Harrison, is trying to explain how carbon monoxide is linked to the pathogenesis of multiple sclerosis.
Hello, my name is Dr. Ines Ramos Harrison, and I'm going to present data from an ongoing project investigating the impact of carbon monoxide exposure in the brain. Whilst the impact on neurons, here in yellow, is perhaps the most studied, I'm actually interested in CO's ability to modulate glial cells like astrocytes and oligodendrocytes, here in purple. My research focuses on multiple sclerosis, which is the most common neurodegenerative condition affecting young adults, and MS is characterized by inflammation, axonal damage, neurological dysfunction, and myelin-producing oligodendrocytes are especially vulnerable. This results in demyelinated lesions in the brain and spinal cord that lead to the neurological symptoms experienced by patients with MS. I appreciate that some of these terms might be scrambling your head if you're not from this area. So to give you a very simplistic analogy, myelin is that fat insulating sheet surrounding the axons of neurons, a little bit like the plastic protecting a delicate electrical wires, if you ever seen a cut out electrical cable like here in this image. So in MS, this myelin insulation is compromised, which leads to issues in the nervous signaling, which are actually electrical impulses. So for the past couple of months, a talented MSci student, Tabitha Studley, has been investigating the effects of low-dose CO exposure in vitro using a oligodendrocytic cell line. She has been growing these oligodendrocyte-like cells in culture, exposing them to different doses of carbon monoxide for different periods of time, and looking at the overall impact on the cells using flow cytometry. And so far, preliminary results suggest that exposures up to 120 parts per million for up to 72 hours do not impact cell viability, but may deregulate normal cell cycle. And this is quite interesting because although CO exposure does not kill these important brain cells, it somehow disrupts the delicate cell cycle. Now, the cell cycle is essentially the life story of a cell, how it grows, how it copies genetic material, so it's DNA, and how it divides to create new cells. And as you can see here in the bottom right corner, this cycle consists of distinct phases with very important checkpoints along the way. And we're currently investigating this further, but I did wanted to showcase the results in this graph here on the left hand side, where you can see that the ratio of cells in G2 to M and G1 increases with incubation time across all groups. So you can see the ratio is higher for 72 hours compared to 48 hours CO exposure, for example. But interestingly, it's particularly high at 10 and 40 parts per million. With the exposure at 120 parts per million showing a ratio that is similar to that of the control. And similar ratios between control and 120 parts per million, despite different ratios at 10 and 40 parts per million, indicate first of all, a non-linear dose response to carbon monoxide. And because we do not see any impact with regards to cell death, so there is no clear dose dependence toxicity issues, this suggests that the ratio changes are not due to cell death, but they're rather due to CO's effect on the cell cycle regulation. And similarly, we are also investigating CO's effects on the expression of myelin markers like myelin basic protein and the proteolipid protein. And as we seen earlier, myelin is the specialized fatty membrane that insulates the nerve fibers. Is created when the oligodendrocytes in the brain and spinal cord essentially wrap the membrane around the nerve fibers multiple times, like the electrical tape around the wire, and this creates a compact multi-layer structure that could be up to 100 layers thick. And the high fat content of myelin gives myelin its characteristic white appearance. But it's the specific proteins in myelin that keep these multi-layers tightly together. And the two myelin proteins we are currently investigating are MBP and PLP. And as you can see in this diagram here, they are differently distributed across the membrane, with MBP here and here acting a little bit like a zipper or Velcro. They're pulling the different layers together, and the PLP here in purple helping stabilize the whole structure of myelin. Our current experiments are investigating how CO impacts the expression of these proteins through a combination of immunofluorescence and other techniques. And the staining panel here on the left hand side, you can see in green the myelin basic protein, so the MBP, stained in oligodendrocytes cells in vitro across the different experimental settings of carbon monoxide exposure, and results are currently being validated, and we expect to have an update very soon. And similarly, we also have a PhD studentship sponsored by Sheffield Hallam University beginning this autumn that will continue this work and further advance our understanding of how carbon monoxide influences stress responses in oligodendrocytes and astrocytes in vitro, specifically targeting cellular mechanisms that are relevant to the multiple sclerosis pathology.
So I'd now like to introduce Megan, who's going to talk about the impact of low-level CO on the brain at the cellular level.
Hello, I'm Megan. I started my PhD with Mari in October, but I've been working in the CO research group for a couple of years now. My PhD is looking at the impact of low-level CO on the brain as a whole, not just on the cellular level, but today I'll just be showing you kind of the data that we have so far on that cellular level. So as has already been mentioned by quite a few people, carbon monoxide has this effect on cells. It can both be protective and it can be toxic. And we know that carbon monoxide can result in the changes that it makes. We know that that's due to its impact on the electron transport chain. So the electron transport chain is the third process in something called cellular metabolism, which Tripene talked about, and specifically within the electron transport chain, carbon monoxide targets cytochrome C oxidase, which is a key enzyme within the electron transport chain that creates ATP, and without ATP there's actually no cellular energy. So cells can't go through metabolism and therefore can't progress within the cell cycle, a bit like what Ines was telling you about just before. While we do know that this is the process of carbon monoxide's impact, we don't actually know the direct impact on cells. So we know that this happens within a system, but we do want to know kind of at what levels can we characterize them as being protective and at what levels can we characterize them as being toxic. So the way that we're going to do this is by 3 different steps. So the first will be on the viability of cells. And as Ines has said, and Samarine, we've already done some data on those viability, but mine at the moment is looking at astrocytes, because astrocytes are a key cell in the brain as they regulate gaseous exchange. So being able to look at them, we know if there's a change in those. We also want to look at the death mechanisms. So there's different ways that cells can die. And at the moment, the literature shows that it's apoptotic in nature. So we just want to make sure and kind of confirm that that death mechanism is being seen. And then the last way that we'll do it is by looking at metabolism. So a lot like triphenol, but just seeing how metabolism changes throughout those cells. So the first piece of data I have to show you is just on viability. So a bit like Ines said, there's no kind of linear change, and there isn't actually a clear toxicity, and they all seem to have quite similar toxicity levels. And we are testing out using a 400 parts per million dose at the moment, but kind of that's preliminary data. I didn't want to show you data that I've only done one run of. So hopefully in the future you guys will be able to see that data. This is done on the U87 cell line. So again, it's astrocytic, but it is a diseased cell line. Because as a start of your project, you're kind of learning how to do everything. So we needed a robust and easy to use cell line. So that's why the U87 was a good choice. We then looked at the death mechanisms on these U87s. So we were able to characterize early stage apoptosis, late stage apoptosis, and necrosis. And as you can see, again, there's kind of not really a change in trend, at least not a significant change. This is, again, we will try out 400 parts per million and see what we see there. But again, that's preliminary data. But as so far, this doesn't seem to be a toxic level as of yet. And then the third is looking at metabolism. So here there's 2 cell lines that I've been able to show you. It's the astrocytic one that we looked at in the first place. And then there's also a HUVEC, which is an endothelial cell. The reason why we're looking at endothelial cells is because endothelial cells line the inner capillaries of your blood-brain barrier. So we can see how carbon monoxide can impact healthy cells on the blood-brain barrier, which is really important when you're thinking about overall brain health. And as you can see, those diseased cells don't seem to have changed. Again, a very linear progression on metabolism. And at the bottom, you can kind of see those healthy endothelial cells. There is a little bit of a dip in the middle, which would kind of, we hope that it's we're kind of finding the toxic and protective effects, and that there may be a lull in there. But again, this will require a lot more research in the future, but it's really cool data as for now. So what is next? I've only shown you a little snippet of the data that we have, but we do plan to do more of the endothelial cell analysis. So those HUVECs that I talked about, we want to do the death mechanisms and the toxicity levels. We want to make sure that we buy some primary human astrocytes that are healthy, so we kind of know how carbon monoxide impacts healthy astrocytes, not just diseased ones. And then finally, we want to look at the cytochrome C oxidase, to kind of see if that is actually being targeted at these really low levels. And then obviously all of this will go on to inform my second work, which will be looking at humans. I'll be doing human studies and kind of be able to tie these 2 together. I'll be using ultrasound and electroencephalogram, so electrical activity, and being able to tie both the cellular and the human part will be really interesting. And I think Mari's going to give you a little talk now about the ultrasound and the other human work that we do within the research group.
Thank you, Megan. And I think that leaves us with the last segment of this, which is all about the adult brain and behaviour. And the person who really should be presenting this is Lucy Wilson. This is part of her PhD work, but I'm afraid you are stuck with me. So hopefully that'll be fine. So the thing that we do here at Sheffield Hallam, which is kind of unusual, and is how I got into CO in the first place, is that we're actually exposing people to CO and seeing what it does to their brain activation. And this probably is a little bit less dramatic than it sounds, because we're giving very, very low levels, pretty much on par with what you'd end up having as exhaled CO levels if you were walking next to a busy road, just with more monitoring and less chance of getting run over. So what we do, we image people with transcranial Doppler ultrasound, seen here, and also with MRI. And we got a setup that allows us to deliver CO quite carefully and also safely to the participants. It's usually the same way for all of our studies. So we've got your participant here breathing on a mouthpiece, breathing room air, and at some point we will introduce carbon monoxide into the breathing circuit, and they'll breathe that, and a series of one-way valves will ensure that this is the only thing that will they breathe all of the carbon monoxide as part of the study. It's a nice setup because it allows us to do this remotely from a distance, which is the case of the MRI, but it also allows us to do it at without the person being aware of what is happening. And that's important because we obviously don't want them to know whether they're getting CO or whether they're getting air control. So typical setup will be, we do a baseline measure, we give them a breathing intervention, so exhale either CO or air if it's controlled, and then we do the measurements again, so we can compare baseline to the post-inhalation measurements. And that way we can get a really robust idea of whatever measurements we're interested in, how it's affected by CO. So that's the sort of stuff that we do. So I'd like to first show you what happens to the brain response at rest, and that is brain blood flow velocity. So not brain blood flow in general, but brain blood flow velocity, and you can see it here. So the vessel we're targeting, and this is with ultrasound, is the middle cerebral artery. It's around here. It's a great vessel to target because it's kind of large, easy to find. It's a big and important vessel in the brain, and it's also common. It's a common site for strokes, which is linked to CO exposure. As a matter of fact, one of the studies that highlighted this link between CO and stroke was done here in Sheffield about 15 years ago, so it feels quite prudent to be targeting this particular vessel indeed. And here you see the brain blood flow velocity going in centimeters per second here, and this is at baseline, so I'll put in a line here for baseline, and then we've inhaled, the person here has inhaled CO, and as you can see, after 1 minute, and 2 minutes and 3 minutes, this velocity profile goes down. And you can see it goes in step with the heartbeat. So it doesn't necessarily look like the heartbeat is changing, but the velocity is going down. So that's just one person for illustration. Here we've got our group results, which is less colorful, but shows exactly the same inverse relationship. So the higher your exhaled CO, the lower your blood flow velocity. So that all shows one particular thing. I should add that these levels of velocities are all within the standard physiological range, so we're not changing things to a sort of pathological state here. We think that the reason for this happening is that there is a vasodilatory response. So CO can cause vasodilation, probably through nitric oxide, and we assume that this is indeed the case. So if you have a vasodilation response, so generally you get an increase in blood flow, but the blood flow velocity will be inversely related to the diameter of the vessel, and therefore the velocity should go down. And I think this is what we see here. So Lucy has collected blood samples of all of these people, and we'll analyze them to see if we're seeing a change in nitric oxide profile. So hopefully I'll be able to tell you whether our hypothesis here is correct very soon. So why should we care? I mean, we are changing a little bit of blood flow within normal physiological parameters. I should mention that this is just due to a raise in exhaled CO for most of these people with about both 1 parts per million. So it's a low dose, changes blood flow velocity a little bit. Why do we care? Well, apart from the fact that changes in flow is linked to certain diseases of the brain, such as vascular dementia and stroke, indeed, there are more immediate effects that I'd like to highlight in this talk here. So first of all, we got, in addition to all of our imaging data, we've got brain response, or behavioral responses for all of our participants. And this is ranging from simple reaction time tasks. So this is a response processing speed, simple one, to more complex response inhibition and cognitive tasks. And all of these are telling the same story. They are telling me that people's performance are getting poorer with CO. So just looking at this here, you have the reaction time here, simple task, no change in the air protocol, but a significant increase in reaction time. So that's a slowing of the response post-CO. We didn't really see anything in the go, no-go tasks, but I'll get back to this once we talk about the MRI images for this particular task, because we've got MRI images for all this. And then we've got this latest addition, which is a more complex, pretty much diabolical task, which is called an NBAC task, where you have to remember a letter from 2 steps back. And I can tell you, it does get tricky quite quickly. And again, we're seeing the same thing here, that there is a slowing in responses in the CO, but not in the air. I should mention that these two here is 3 parts per million rise in exhaled CO, and this is just 1 part per million rise in exhaled CO. So these are very low doses. So what do we actually, so we've got a change in brain blood flow velocity, and we've got a change in behavioral measures. So I think what we need to do is just move on to the pretty pictures. So this is the brain activation for the first task, this one mentioned here, where people were responding a bit slower to simple tasks, pretty much saying, pressing a button when a red dot appeared on the screen versus when it wasn't there, with the gap varied so that you didn't know when it would arrive. And what we saw here in the brain activation maps afterwards was that there was a higher activation following CO inhalation compared to air. It was mostly on the left-hand side of the brain. Do not confuse this, because we typically flip these images in neuroimaging analysis. So the left is right and right is left. I know it's confusing, but it's typically on the left-hand side of the brain, but it is slightly bilateral in nature, which is very good. The left-hand side means nothing really. It just means that they were doing the task with their right hand. So that's usually how the brain works, it crosses over. And the area that we saw the higher activation in are areas associated with motor control and attention regulation. So the premotor cortex, the primary motor cortex are all about planning, preparing, initiating movement. And then you've got things like the posterior cingulate gyrus, which is attention regulation. The activation here went into the precuneus, which is all to do with movement, engagement, and memory. We've also got the frontal gyrus involved here with attention and decision-making process. And all of these are labeled on this slide here. But this question, when I saw this, was why is there an increase? Because previously we have seen a reduction in activation with simple tasks like a breath hold or a visual stimulation or a finger tapping. And we always assumed at this point and we still do, that this was to do with the brain blood flow changes that we saw earlier. So effectively, the way that MRI works is that it looks at the ratio of oxygenated to deoxygenated blood flow and looks at the changes in these. So we assume that when a brain area is active, it would require more oxygenated blood, and therefore that blood will go there. So that's why we always do an MRI task with a rest, where you do nothing and you measure that, and then a period of activation where you do something, and ideally blood will go to that part of the brain. So you look at the contrast between these two. In this case, for this part here, the activation would be the sensation that, oh, there's a red dot on screen, I need to press a button. So that is how we decided. And if you then predilate your vessels, you have less room for increasing the blood flow to that part of the brain, so you'll get a smaller difference and a lesser signal. And that is what we assumed was happening with these, and we still do. But here, there's something else happening. And I think this is neurological in data because it's based on behavior. And what do I mean by that? Well, there is a potential learning component to this. So typically, regions involved in task processing before training can show reductions in activation after training. So many decreases in activation can be linked to improved task performance. What do I mean by that? Think about it in terms of learning how to ride a bike. So when you learn how to ride a bike, the first time you're riding your bike alone, you wobble, your brain is very much focused on what it needs to do. And it cannot, you can't fall off, you can't do anything like that. When you get good at it, you think about what to have for dinner. So when you're an adult, and this is the thing, so the brain invests less energy towards it. And this is what we thought. And if that's the case, we would see a reduction in activation in the AIR protocol, but not in CO, that's this learning component. And indeed, this is what we did see. So we assume that there is a learning component here. This is a robust change. The activation pattern follows the posterior cingulate, so here, about here, over and all the way into the sort of the precuneus area here, meaning that it goes from attention regulation into control of movement and memory. And that is important because it means that it's just not a single blob and we can actually tell what's happening here. We can tell it also because we've modeled this against the behavior. So we know here is an example of a model that tells the computer when the person is seeing a red dot and how long they take to respond, and tells it to look at the activation associated with that particular model. So this is direct link between the behavior and the neuronal activation. So that's our model design. So we know that this is to do with decreased attention and vigilance. We see the same in the go and no-go task, but here, since we are choosing whether people click for an X or click for OK, because that's the task, click for an X, do not click for OK, we can contrast whether people are actively doing something or inhibiting doing something. So, my goodness, I can't press its K versus, oh, I have to press this next. And that means we can tell whether it's the inhibition activity that is influenced by the CO or the action that is inhibited by CO. And in this case, it's the inhibition. So we can see that people are just using more brain resources to deal with inhibiting, not pressing a button. And that is also possibly why we're not seeing any significant results here in the baseline, because we can't really do an average of things not happening. We can only measure what happens when they actually do press the button, and there we don't see that it affects it. So just to show you an overlap of these results, because they're very similar responses, just they're linked to the same regions, just different areas of the same regions, if you know what I mean. And we're seeing that there is a great deal of overlap between the reaction time task, which is shown in red, and the inhibition task, go, no-go task, which is shown in blue. And that leads further credence to the fact that these regions are indeed involved. So both of these are increased with CO showing a possibly reduced learning effect, bearing in mind also that these are linked with a poorer performance on these tasks. Again, some common activation areas. So where does that leave us with that very difficult test? Well, we don't have the fMRI data there yet, but what we do see, so this is where you have to remember a letter from way back, or 2 steps back, it becomes tricky, I promise. We see a difference behaviorally, and we see a steeper or stronger relationship with blood flow during this task than we do at rest, but we don't have the fMRI data yet, so that's pending. We'll see if that shows the same as the previous 2 tasks, but my guess is that based on what we're seeing already, it probably will. So in summary, because I know we're running out of time, for this part, what we're seeing is that brain blood flow is affected by CO doses that can be as low as raising XLCO by 1 part per million, and this is in healthy volunteers. The impact is dose dependent, possibly task dependent, and it could mean that we have impaired cerebrovascular reactivity, which is a marker for disease development or increased disease risk. For the MRI, we show task-dependent impact of CO at doses that are raising XLCO by no more than 3 parts per million. Again, very little, below what you get of a smoker, and again in healthy volunteers. And whilst previous research has shown a decrease in activation for physiological tasks, the new data on the cognitive task shows increases in activation, suggesting that there is indeed a dual function of CO on the brain, both related to blood flow, which has disease links, but also related to neural function, which is behavioral, but also, again, could relate to disease. So two different ways of influencing the brain. This increased activation that we found could indicate that there are decreases in attention and vigilance, even at very low doses, and the behavioral data supports all of this. So we're seeing diminished cognitive performance after CO inhalation. So that's the end of that. And just to summarize the whole talk for everybody, I think we've agreed upon these points here, and that is that CO, even at low levels, have measurable effects on brain development, cellular functions and pathways, and behavior and brain activation. That the impact is not uniform, neither across cell types, doses, nor experimental paradigms, although we do see some parallels, and that CO, even at very low doses, impacts the organism in both health and disease, and it does so in different ways. That only leaves me with a few things to do, and that's to sort out through the acknowledgements. We'd like to thank Celine for all of her help with all the lab work. Pretty much any lab work that you've seen in this talk here has been supported in some way or form by Celine, and we'd be lost without her. Also Sydney Peary and Louise Austin, who had been project students working with Samreen on her cancer research. We'd like to thank Marisha Placek at the University of Sheffield for all of her continued help and support. Also Isabella Myers for being a valuable team member, and of course, last but not least, Adrian and Gerarda, Kimberly and Lauren and Court for their continued support of the work that we're doing. I'd like to also thank the team, myself, and Simon has kindly given us a picture of everybody here. I'm not sure who I'm in this, but my guess is probably Granny. But in case you can't recognize us, here is a more recent picture that Adrian took in November, and I've included Simon here with his permission so he won't feel left out. If you are interested in our research, we do have a social media platform run by Trifine, as I mentioned before, in collaboration with Megan. So come find us here if you're on TikTok or Instagram. And again, please submit your questions to Lauren or in the chat in the 1 minute that we have left of this talk. I'm really, really thankful for your attention. Thank you from all of the team here for listening to what we're doing, and I look forward to hopefully working with a lot of you in the future. Thank you very much.